Okay, so I guess it's time to start. And I would like to thank all the participants to join our Accela webinar. Today we have a very exciting new technology to introduce. Is the beacon discovery from the Bruker cell analysis. And I have the pleasure to introduce you to Tiago Santos, which, correct me if I'm wrong, you are the manager, business development manager, right, at Bruker Cellular Analysis.
Yeah, I'm
So
part of
basically,
the
you are the
marketing.
right
Yeah.
person to give us the good vibes about this new technology. Yeah, and I'm Riccardo Pasculi from Accela, so I'm taking care of the application support for the cellular product, and I'm very excited to learn more about the beacon.
Absolutely. And
Thank you,
thank you so
Tiago.
much for that introduction, Ricardo. And yeah, I'm really happy to be here today and really grateful to Accela, to Ricardo, to Igor, to the team for allowing us to be here and to present to you this technology. And so the way that I separated the technology, because Beacon itself, as you will see, it's a multi-application platform. So first I'll go over what makes this platform at the live single cell truly unique in the terms of the technical details, and then I'll go to two different applications, and we then can open for Q&A and discussion if you have any questions or ideas or even really talking about your specific projects. So yeah, I'm Tiago, part of the market development team at Bruker, actually Bruker Cellular Analysis. And today I'm going to talk to you about a technology that is trying to unleash the power of live single cell analysis in many different areas, in immune cell profiling, cell therapy development, antibody discovery, and so on. And so if we look at the current technologies available, what Beacon is trying to do is really revolutionize the way that we look at functional screening of live single cells. Because in these types of projects, especially immune cell profiling, of course, most researchers are really looking for depth of knowledge in terms of how is the cell responding, what cytokines is it secreting, what surface markers are there in that cell, and also transcript base as well, try to understand the response of that cell. The thing is, you have to truly separate the sample, split the sample in order to go to different platforms to do so. So, for example, you can use ELISpot to look at cytokines. But the thing is, it's a snapshot of that response. You basically are looking at already secreted cytokines. Or otherwise, you can use flow to look at surface markers or intracellular cytokine staining to look at cytokines inside. But once again, you're trying to predict if that cell is responding or not. You're not really seeing true response of those immune cells, for example. And on the side of transcripts and RNA-seq, transcriptomics in general, of course, it does provide you a lot of data, but at the same time, the correlation to protein and to function is far from one-to-one. So you may question then, if you want to really focus on a specific cell, how can you know what is that cell specifically doing? And how is this data actually, how are you able to actually bind together all of this data in terms of the aspect of robustness of data? Because you're looking at like very different platforms in order to get all of those functional parameters. And how as well does that cell behave across time? Because these are very much snapshots, time points where you have the data in terms of cytokine secretion or cytotoxicity, and then that's it. That's the data that you have. And that's where Beacon really is trying to revolutionize the way that we look at functional screening by providing a way to not only looking at live single cell, providing the way to do co-culture, but providing you the way to look at temporal dynamics. So not just a snapshot of a given parameter of function, but really tracking what the cell is doing. And so as a teaser, I'm going to show you one well. The well, we call it in the beacon, we call it a nanopen. And so in this nanopen, we have selectively loaded live single cells. We have selectively loaded CAR T cell that it will appear in green. We have loaded a target cell that will appear in blue, a cancer cell, and an off-target cell, a healthy cell in pink. And now as you can see through Beacon, in this nanopen, we see the interaction, the contact between the different cells. And as you see, the green cell did the contact with the target cell, with the cancer cell, and actually it changed the color. We analyzed the fluorescence of caspase-3, an apoptosis indicator that allows us to really track exactly when that specific CAR T cell did contact with the cancer cell and when it was able to kill it. But not only that, as I mentioned, we have loaded also in this nanopen an off-target cell, a cell that is healthy, and in this way we can actually track if that CAR T is specific for other targets and is not actually going to kill other healthy cells and off-target cells. So that's just a bit of a glimpse. I will show you as well other functional assays and really the data that you can get out of the Beacon in this type of assays. But as I mentioned, Beacon is really leading when it comes to single cell functional analysis in very different applications. So in antibody discovery, which I also will mention during this presentation, it's the world leader really when it comes to functional screening B cells and finding those antigen-specific antibody secretions for even for hard targets in a very reduced timeline. And also it provides you multiple functional screenings to profile those antibodies. In cell line development, I won't focus too much in cell line development during this presentation, but it's also the gold standard because as you will see, it allows to culture the cell lines in very good conditions, and it is really the gold standard when it comes to guaranteeing monoclonality, above 99%, guaranteeing as well the capacity to look at tighter ranking of the clones and also population dynamics and the stability of those clones. And then I'll focus most of the time actually in the immune cell profiling application, where really Beacon has enabled researchers to look throughout the pipeline, throughout discovery, validation, and translational, at the potency of a given cell product, at the capacity to understand if it is going to be effective or not in the patient, even providing biomarkers that correlate with patient or non-patient response sometimes. So yes, during this presentation, I'll focus on antibody discovery and also immune cell profiling. We call it T-cell, but I also want to let you know that the platform is quite flexible in terms of the types of immune cells that you can analyze. So what's Beacon? Beacon is an optofluidic platform. It combines optics and microfluidics, but more than that. As you see in this image, within this platform, you have multiple instruments. And all of these instruments are combined together and fused to really process the cells and analyze live single cells and do multiple functional assays. So the Beacon was actually launched back in 2017. And since then, it is routinely used by the top 15 pharma, CROs, especially in antibody discovery and cell line development. And more recently, we see more and more academic institutions actually using Beacon for other types of projects in cancer immunotherapy, infectious diseases, autoimmune, because of this level of functional assays that we have at a single cell level. This is just the number of instruments worldwide. What I really want to focus on is really the increased number of super high impact factor publications that show the value of using this data in very different types of projects. And we have plenty of KOLs worldwide using the platform in these different applications. For example, Professor Stefan Eichmuller for the discovery of new TCRs, screening on Beacon, and Professor Antonio Zamora in development, validation, and translational workflows using CAR-T in the clinic and trying to find those biomarkers of response to predict response of patients based on the potency of the cell products. But also leaders in antibody discovery and cell line development using the platform to do so. And so I'll go now a bit more in detail into the technical aspects. How has Beacon actually allowed these researchers and others to find this valuable data? First, you have a deterministic control over the single cells that you're working on. What I mean by this is that when you load the cells into the chip, then you can, based on morphology, based on surface markers, really select the cells that you want and load them in the nanopens, in the wells that I mentioned, in a selective manner, which means you're not bound by random capture of cells, by Poisson distribution. You yourself are selecting the cells that you want. Then you have the highest parameters on chip, and I will talk a lot about different functional assays. because we can culture the cells. We can maintain cells in a proper state, alive, which means you can conduct multiple functional parameters. You can culture the cells during hours, days, well, even weeks, but really depends on the biology of the cell, of course. And then we also mentioned infinite possibilities of chip, because after collecting all of the functional data, you can retrieve and export the live single cells that you just assayed for outside of the instrument for downstream analysis, being either expansion of the cells or really going to a transcriptomic workflow, TCR sequencing, RNA sequencing, and so on. So that's why we call the Beacon a functional first platform. You first try to screen which are the subsets of cells providing or showing that functionality, and then you retrieve the cells for downstream analysis. This way, try to bridge the gap between both omics. And also, it's fully automated, as you will see, even in the loading of the reagents, loading of the beads and cells. It can be fully automated by the Beacon, and it's scalable. You can screen thousands and thousands of live B cells, for example, but also if you're working on a small subset of antigen-specific T cells, for example, you can sort those within the OptoSelect chip. We call it also OptoSelect device, where you can really sort even cells based on specificity, which means that even if it's a rare subset, you can also work on that. So this is Beacon. It's actually an end-to-end solution that goes from the instrument to the software. So you see multiple different instruments here. As I mentioned, the first Beacon, the original Beacon, was launched back in 2017. It's actually that one, the dark Beacon in the back. The original Beacon, it's a big instrument, I have to say. It's like 1 meter 8, 700 kilos. It's a big platform. And so especially from academia, from universities, researchers that really wanted to have access to this type of platform, it was actually a challenge, an obstacle, the size of it as well. And so during the last 2 years, we have actually put a lot of efforts in developing, and we just launched this year the new Beacon. As Ricardo mentioned, the Beacon Discovery that is here. It's actually quite smaller. It's a benchtop platform, more compact, more accessible, that enables more researchers across the globe to have access to this type of technology. And honestly, the biggest difference between all of the different varieties of the beacons, it's not the base of the technology. The technology inside of the beacon discovery is the exact same as the one used by the big pharma and 160 customers. The difference is really the throughput, because if the original beacon can run 4 chips, that I'll mention after, the beacon discovery is really focused on one nest, the capacity of analyzing 1 chip per run. But all of the same, stays there in terms of the power of the analysis of functionality of live single cells. So now you see here the consumables, you see the chips, the OptoSelect chips, we call it as well OptoSelect devices because they are really unique. They are microfluidic devices that really enable this type of technology to work. And it's really where the magic happens. And so you see here different numbers, these tied to different chips, and they vary depending on the number of wells in each of those. So for example, for cell line development, we recommend the 1750 chip, which has this number of wells. But for antibody discovery, it's on the 20,000 because you need to increase the throughput in order to detect those antigen-specific antibodies secreted by B cells. And in the middle, it's the sweet point for actual immune cell profiling. And so we recommend this one to do co-culture of immune cells with target cells and do the multiple functional assays. But this also means that in a single chip, you're analyzing more than 3,000 live single cell reactions, which, as you'll see, provides actually a lot of data. And regarding data, you also have a complete UI and data analysis integrated in the platform. You have an experiment wizard that we have developed and launched with Beacon Discovery that it's actually very intuitive and easy to use in order to plan your experiments, even the timeline, the agenda in terms of loading the beads and the cells, and all of the assets that you want to do. And then you have two major software analysis integrated in the platform, Image Analyzer and Assay Analyzer. Finally, I'd like to mention as well that the scientists actually, and this makes us very honored, the scientists considered and gave this award to Beacon Discovery this year, considered one of the top innovations of 2025, which of course makes us very proud of it. And hopefully more and more researchers across the globe will be able to have access to this type of technology. Because the magic, as I mentioned, actually works inside of these microfluidic devices. As I mentioned, these are very small chips, the size of the palm of the hand. What you're seeing here is a photograph zoomed in into the chip, where as you basically can see like corridors, multiple corridors, and then you can see like small wells, small nanopens. This is actually the 20,000 nanopen chip. And these are the wells that basically will be able to sort and analyze live single cells. So imagine that you're working with your sample. We have, of course, standardized protocols and also a team of field application scientists to work on developing and supporting you in your workflow. You're preparing the sample, you're loading the live single cells in suspension into a well in a 96 well plate. You place the 96 well plate inside of the beacon, and then using microfluidics, now you see the representation in this video, we are basically pulling the sample into the OptoSelect chip. What you're seeing here, loading the sample into the chip, and then the cells will flow within media, because we have a perfusion of media across the corridors. And now what we'll see is some sort of a light cage that goes around the cells and selectively picks each of the single cells and loads them in each of the nanopens. And this is tied to what I mentioned regarding one of the major innovations of the platform. You have deterministic control over the single cells that you want to analyze. So how can we do this? The thing is, the chip is constructed in a way that has like millions of phototransistors within the chip, and so we have a light in the instrument that precisely hits each of these points in the grid and allows us to create a light-induced mild electric field. And we call this optoelectric positioning, short as OEP, our technology that allows us to actually move cells, particles, bits, and be really selective on the types of cells that you want to work on. So this video is actually taking out of the beacon. This you can actually see happening live on the beacon. You have this mix of population, a mixed cell population within the corridors, and then we ask the beacon to basically select one cell per each of these and load them in each of these nanopens. And of course, as you can see, each of these nanopens is numbered. So every single assay, every single functional assay, every single data, every single movement that will happen within each of these nanopens will be recorded, tracked, and you'll have access to that information, all linked to that specific single cell. And this will tie to the robustness of the data and the level of functional assays. To showcase the power and the control that you have, in this video, we made basically a path for that cell to go. And so Beacon created, once again, this OEP, this light-induced mild electric field, to move the cell across this path. And this really truly shows you that at the level of single cell, the control that we have to move cells and to select cells that we want to analyze. Of course, this is just to represent. Then what Beacon can do is do this in a massive high throughput manner in selecting hundreds and thousands of different live single cells across the chip and load them in each of these nanopens. So imagine that you're loading here an immune cell, a T cell, and now that you want to load a cancer cell or an NK cell, you would just flush away the cells in the corridor, bring new cells, and do the exactly same thing for the loading of the cells. And this is also enabled by the second major innovation that is actually one of my favorites, is the fact that the cell is not encapsulated. The cell is not in a droplet. The cell is in medium. The cell is inside of these nanopads. And so throughout the chip, the corridors that you saw, we have a continuous perfusion of media. We have a laminar flow, which means that if the cell is alive within these wells, within these nanopads, of course, it is alive, so it is releasing waste, but also it needs nutrients, it needs oxygen. And so using this laminar flow, fresh media, we can replenish the cells with that and maintain those cells in a proper state to do the functional screening. And as you can see here in this video, just to exemplify, we loaded thousands and thousands of fluorescent nanoparticles, and they are not majorly disturbed because of the laminar flow, which means that a cell with a higher kilodalton would just not get out of the cell of the nanopen because of the laminar flow. And so in terms of the workflows that I will talk about next, they mostly go for these 4 different sections. First, this ability that you have to select the cells that you want, live single cells, and load them in each of these nanopens. Then you can culture them. Of course, this really depends on the type of assay. This would be mostly for cell line development. But this is to mention that the technology is flexible. You can control the media that you use, the temperature, and other conditions that you might want to change. And then you can do multiple assays. You can bring other cells, you can do co-culture, as we will see in the next few slides, and record all of that functional data. And why are we recording all of that functional data? It's to tie to the first slide. It's to get that robustness, functional, temporal data from the response of those single cells to identify which ones are actually the best cells to then, in the fourth stage, recover them for downstream analysis. So these are the main concepts of the technical details of the technology. I will now focus on the immune cell profiling application and really how Beacon is advancing cell profiling in cancer research. And I'll give you as well some examples, case studies and testimonials of researchers working in this field. So first focus on the validated and workflows that we have developed. The first one that I want to show you, we have already had a glimpse at the beginning of the presentation. It's this temporal dynamic cytotoxicity. It's this capacity of loading different cell types, different immune cells and cancer cells in each of these nanopens, and track exactly the contact between these cells and if the immune cell is capable or not of killing those cancer cells. And even more than that, you can actually track how much time each of these immune cells took to kill those cancer cells. Together with that, you can do multiplex cytokine secretion. We add beads. We basically do sandwich ELISA in each of these nanopens, and we can detect up towards 3 different cytokines secreted by each of the single cell. Also, surface markers. You can also detect surface markers, do surface marker phenotyping on the beacon, and a fourth assay that we are just releasing now, and we are super excited to talk about, is actually the ability to track motility in cell-to-cell interaction. So we have integrated in the platform an AI ML imaging software that enables Beacon now to really precisely track the position of every single molecule and every single cell within each of these nanopen. And in this way, have a time plot of the interaction between the cell. When did the cell make contact with another one? When it happened, an increase of brightness of fluorescence of potentially linked to killing, when they disattach, and so on. All of this now is also Beacon is also able to provide you in terms of functional data. These assays, sorry, I jumped too fast on this one. These assays can be done in a multiplex way or a sequential way. You can pick what type of assay you want to perform, and then you have a breadth of graphs of data, table data, assay data that shows really the response of these cells. For example, cytotoxicity heat maps to really look at the different subsets of cells in terms of killing capacity, look at relative quantification of the secretion of the cytokines per nanopen, per cell, of course. Also, phenotype scatter plots, and what I just mentioned in regards to having a time plot that really shows you the interaction between the cells within each of these nanopens. So first, let's go to surface phenotyping. As I mentioned, you yourself have the control over the single cells that you want to analyze. So you can sort cells based on morphology, but also surface markers. What we have in this video, it's a part of the chip where basically we asked Beacon in this mixed population, to find one CD8 positive, CAR positive T cell. So you go through the fluorescent channels, and we have 4 in the Beacon Discovery. You will find one that has these markers. He will select it and load them precisely in one nanopen. Of course, this can be done in a high throughput manner, but it's to show that even if it's a rare cell or a rare subset of cells, he will find those and will load them specifically in each of these nanopens. Once again, they are numbered, and everything that is happening after you will track. So what other functional assays can you do? As I mentioned, cytokine secretion, And I also mentioned that the beacon is fully automated. And you see here exactly that. These are just a couple of nanopens in the larger chip, but it's to show you that we are loading here the third batch of bits to do the cytokine secretion assay, look at three different cytokines. So overnight, he will basically load off all of the different batches of bits. So we flush in the first batch, select one bit per nanopen, load that, flush away, load the second, load the third in a fully automated fashion while you are at home. So when you come back to the platform, actually, you're going to do like some proper science, which is, of course, loading then the live immune cell. In this case, we are loading a T cell. Once again, using OEP. This some people also say this is like a tractor beam that really allows to move cells inside of these nanopads. Uh, Sorry, I hear a bit of a background noise.
Yeah,
If you
sorry.
could please mute. No worries.
Exactly. Please mute yourself. I don't know who is making noise in the background. I'm trying to find who has the mic on. Just a second.
Good.
Okay. All
No
good.
worries. Perfect. So in
Keep
the
going.
third video, what we are actually seeing is the third step of the assay for cytokine secretion, which is loading the target cell. We actually loaded a non-stimulated T cell, and now we are loading the target cell. And so using, once again, the OEP, this technology, we can load this target cell, as you can see here, so close together to the T cell to promote the contact between them. So if it is a CAR T, if it is a T cell and NK that is actually a responder, it will secrete those cytokines that then it's an automated, basically an ELISA sandwich in each of these nanopens, because basically we are capturing the protein by the primary antibody bound to the bit, and then we have a second antibody with a fluorophore that will bind and we'll see the secretion. It will see the fluorescence of the presence of these cytokines. And we can do this in a multiplex way across the thousands of different single cells and nanopens. Second major assay is, of course, this dynamic cytotoxicity assay that looks at temporal capacity to really understand the response of those immune cells. In this case, we are looking at one nanopen. We are looking at bright fields and then two fluorescent channels, FITs and PE. So the cancer cell actually comes in, we bring it in with a cytosolic dye, but we also can track caspase-free indicator. And so what we have done is selectively loaded one cancer cell and one T cell. And then we can track through fluorescence exactly the moment that that specific CAR T was able to kill the cancer cell. And so, as you can see here, it says low cytotoxicity. And why is that? What is the comparison for us to say that? It's because we are doing that in a multiplex way across thousands of different live single cells. So some of them will kill much faster, others will kill slower, others will even be able to kill multiple target cells. That's one other thing. You can co-culture multiple target cells. It doesn't have to be just one. You can also look at other markers like exhaustion markers, try to understand why some cells do not respond well, why they have no cytotoxicity, and even try to understand the causes by T cell death. One researcher actually in events in Slovenia, in Ljubljana, actually mentioned that one of the key things is also not just looking at the capacity of killing, but after the CAR T killed the target cell, understand if it is able to be motile and go and kill another cell. That's super important to really see if it is persistent, if it can resist then in the patient and resist exhaustion. The fourth one that I'm super excited to talk to you about, because we are just launching this, is motillion cell-to-cell interactions. So in this case, in each of these nanopens, the blue marked cell is actually a T cell, and the green one is a cancer cell. And as you see, because we are once again using that AI ML imaging software, it's quite cool, enable this type of tracking across thousands of different nanopens. There's no major contact between these different T cells in blue and the cancer cell. In this 1671 nanopen, actually there is contact in the very beginning here, but there was no cytotoxicity. So it was not a cell that made contact, it was able to kill. What's really cool now is actually looking at the nanopens where we find T cells that were capable of killing the target cell and how fast they did and how fast they moved within the nanopen and how fast they made contact with the target cell. And as you can see here, the 458 actually is the cell that has the pole position because it was the fastest one on actually making contact with the cancer cell, but also killing it. And then we have basically these time plots that enable us to understand better the motility, the cell-to-cell interaction of these cells. So this is a time step. Each of these time steps equals to 15 minutes each. Then on the left of the graph, you have a cytotoxicity that is based on the max median PE brightness that is linked to the apoptosis marker, caspase-3. And on the right, you have basically targets APC, so the cancer cell and the T cell distance. And we consider that there is contact between the cancer cell and the T cell if they are at 20 microns or below. And as you can see, once again, thinking about the cytokine secretion assay, we place the T cell really close together to the cancer cell right at the beginning. And so there's contact. But as you can see, then there's separation. The T cell actually moved away. So yes, there was contact, but there was no killing at this moment. There's no increase of fluorescence of the caspase-3. However, at timestamp 15, there is again contact here below 20 microns, which means there's contact between the two cells, the T cell and the cancer cell, and you see a huge spike in the brightness of the caspase-3 linking to the killing. So at this moment, actually, the T cell started killing the cancer cell. And you can even really track how much time it actually took for the cancer cell to die and how much time did the T cell made contact to the cancer cell. And this type of data is actually super valuable to understand the potency of different immunotherapies and cell therapies. Because honestly, there's not a single platform in the market that enables this. The single cell co-culture and the tracking, the motility tracking across time of what is happening really in the biology of the interaction of these two cells, it's only really enabled through this type of technology. So I talked a lot about different functional assays, cytokine secretion, cytotoxicity, motility tracking, surface phenotyping. All of this data, as I mentioned, is collected for each of these nanopens, for each of these single cells. And then you generate a large collection of functional data for each of these specific cells. The cool thing is that you can then cluster, and you can cluster based on the functional profile. So in this UMAP, basically you have clustered all of the nanopens. Each of the data points is actually one cell, one nanopen, and they are clustered based on how similar they are in terms of function, in terms of the cytokines that secreted, in terms of the killing capacity, surface markers, and so on. So imagine that now you, based on your research, based on what your knowledge, of course, you figured out that the best group of cells is actually the orange one. Remember, these cells are still cultured in the beacon. They are still alive there. Imagine this was the best group of cells. You can then basically go back to beacon and say, get me those cells back. Export the live single cells. So because now I want to do expansion, or even better, you can do single cell TCR sequencing, you can do mRNA-seq. And in this way, Beacon is enabling to bridge the gap between proteome and transcriptome in terms of functional and transcript, because the cells that you're going to actually analyze through transcriptomics are the ones that already showed you that functionality. And in this way, you'll be able to reveal the differential gene expressions behind these polyfunctional T cells. And that's really a remarkable, powerful assay and analysis, functional analysis that you have through the Beacon, and has been leveraged by multiple people in the different areas. And so now I'll focus a bit on those case studies and testimonials of people, researchers in the immune cell profiling cell therapy market that have leveraged this platform. So as I mentioned in the beginning, throughout the pipeline. So for discovery, Beacon has been able to, in a faster way, to discover those binding TCRs, functional TCRs, and even qualify CAR-T libraries. In validation, it provides functional data to really distinguish between which of the candidates is more functional, and giving as well researchers the confidence to move forward with a given candidate. And on translational, more and more we see researchers publishing actually this combined data as a biomarker to predict response or non-response in patients. So I'll give you examples. For example, Professor Zenga working on TCRs. In validation as well, Paul Thomas working on TCRs in order to try to better characterize these candidates, and in translational, Professor Anthony Zamora. In talking about Anthony Zamora, he actually published very recently, back in March, the data that he got through the beacon from validation to translational in his CAR-T clinical trial. And one of the highlights of the webinar and the way that he presented the data is that, of course, he has years and years of experience in working cell therapy and CAR-T development, and his workflow was very much based on like splitting the sample, going to flow cytometry, going to bulk cytotoxicity, going to Luminex even to look at cytokine secretion. And the highlight he gave of the platform is this ability to, with just a sample, focus on the single cells to get multiple functional parameters and readouts that qualify better the potency of getting cell products. And he also highlighted the robustness of the data, of course, because instead of going to different platforms, you're collecting all of this data from a specific one. Then Professor Stefan Eichmuller at the DKF said, leveraging this technology really for TCR discovery. And he said himself it was unprecedented, the number of reactive T cells that they managed to detect, because in every single run, he actually detected T cell reacted against antigens and neoantigens. And exactly on his case study, and of course, this is published data, we can also share with you. He's working on myeloma and ML patients. So he split the sample, of course, going once again to the nanopens. And in each of these nanopens, he loaded a patient bone marrow T cell together with the target cell, the patient cancer cell. And then evaluated in terms of killing capacity, in terms of contact between the cells, in terms of cytokine secretion. And throughout the 22 patients, it's listed here, he detected 623 reactive T cells. And from the 19 unique TCRs, actually 85% of them were actually validated outside of the platform, being tumor-specific reactive TCRs, which is astonishing in this field because of how hard it is to actually functionally characterize these type of TCR therapies, especially to neoantigens. And as well, now they have a base library that they can work on to try to validate and move forward then to the clinic. Second, focus on still on discovery, but now and still on TCRs, but even more with an aspect of validation. The work of Professor Zenga is truly astonishing. He's working on a very aggressive solid tumor. That is very heterogeneous, as we know. It's head and neck squamous cancer, and so he had access. He had already screened the tissue from the cancer from patients, and through spatial technologies, he managed to identify the different subsets of cells in the tissue, and he identified some that were potentially the perfect markers, which are the tumor-infiltrating lymphocytes. Those T cells that not only managed to attack the tumor, but manage to get in the tumor and potentially bind to neoantigens, potentially show functionality and killing capacity. So, working on that, he dissociated the samples and he went on Beacon. And once again, selectively, he loaded one of those tumor infiltrating lymphocytes in each of these nanopens, co-cultured with the target cell, and then looked at the binding, the contact between the T cells to the cancer cells, and selected those that showed functionality, the ones that showed cytokine secretion, the ones that showed the killing capacity. For what? Because of what I mentioned. It's a functional first platform, but it's not an endpoint. What he managed then is to pick up that T cell, export it, and do TCR to get the sequence, because this is the type of TCR in these T cells that has been already validated in the discovery setting, because we already see that that specific T cell was able to bind, was able to kill the cancer cell. And so now he is actually super happy because he screened 5 patients in just 3 months. He identified TCR sequencing functional cells for each of these patients, that now he can work on these TCRs to develop a cell therapy that is really personalized to that specific patient. Then, still on TCRs, but more on validation, the case of Professor Paul Thomas, he was, and he published this very recently as well, he was looking at functionally characterizing two different TCRs, and he was not finding a way to do so and actually have the data to show you. He was doing bulk cytokine and cytotoxicity, and as you can see between the two different TCR candidates that he was working on, he couldn't find significant data that would show that one was better than the other in terms of functionality, in terms of activity against the cancer. When he used Beacon, he found that significant data, that's for sure. And actually, one of the TCRs were very much showed a higher profile when it comes really to cytokine secretion and activity of that single cell against the targets. And this is data that was simply missed in bulk cytokine analysis. And now he can even work more on this. Now that he has the cytokine secretion, he also did the cytotoxicity to try to as well functionally validate the killing capacity. And that gives him a lot more confidence as well now to pick up the data and go to the next stage of the development of these TCRs. And that's the type of data that Professor Anthony Zamora also managed to collect now on cell therapy CAR-T products. Working in the clinic, this is actually a super nice publication we can share with you, was published back in March in Journal of Clinical Oncology. He was working on a phase 1, phase 2 study in mantle cell lymphoma. And so what you're seeing here is what I mentioned, tracking really the different subsets of CAR-Ts and the way they behave. And this is only enabled using Beacon and live functional screening, because he managed to stratify the different CAR-T subsets within the products based on the killing capacity and the cytokine secretion. And why is this relevant? So we talk, it's quite common, and we more and more talk about cell therapies and we talk about CAR-Ts, but the thing is, each CAR-T, especially if it's an autologous approach, each CAR-T is very different from patient to patient because the CAR-Ts are engineered with our own immune cells. So depending on the situation of the patient, his immune system response, those CAR-Ts won't always have the same type of response and the same potency. So it's crucial in order to give tools to clinicians, to researchers, to understand from the very initial, the get-go, from the expansion of the products, if those subsets of CAR-Ts are actually having that killing capacity that potentially will correlate with response. And as you will see, it did. Because not only the patients in the cohort that responded had a high level of CAR-T cells that were able of killing fast and killing multiple cells, but also the subjects that did not respond well to the therapy had unfortunately a high number of CAR-Ts that didn't have that killing rate capacity and also had a higher level of CD8 positive T cells with low levels of cytokine secretion, showing that while reading the functionality, the potency of these products early, he managed to correlate and use this as a biomarker of patient and non-patient response. And what's interesting as well in this study is that, once again, using other platforms, using both cytotoxicity, it was non-significant, the data. It was not really showing the difference between these different products because it's simply misses out. These type of technologies doesn't have the depthness needed that the Beacon provides while going to the single cell level, going to temporal analysis of cell-to-cell interaction on a live functional screening. So that's a bit of the details in the case of cell therapy and immune cell profiling. Considering the time, and since I wanted to as well focus on antibody discovery, I'll jump into that because, of course, Beacon was first launched and really focused on antibody discovery, cell line development. And of course, on Beacon discovery, the smaller, more compact, accessible Beacon, you can also do antibody discovery. And that's the flexibility that it shows the instrument for different universities and different platforms. The value of Beacon in antibody discovery, and it's really the leader on that, is changed completely the picture, especially in terms of timelines. Because through a typical hybridoma phage display, you're looking into months and months of work of technicians, of researchers to find those monoclonal antibodies, while on Beacon, it's basically a couple of weeks. And the work of antibody discovery and profiling on Beacon actually takes 1 day. You can see already antigen-specific antibody secretion and binding within like 7 minutes. Plus, we maintain the diversity, as you'll see. It's immunization, isolation of cells, and then we have multiple kits, as you will see to analyze different B cells from different types of samples. So yeah, you start with immunized samples, then we have, as you'll see, end-to-end solution, different kits to isolate and work on those B cells. Then once again, you selectively load those live B cells. You do the assays. I'll go a bit deeper on that. You recover the B cells, or you can do, you can capture as well the sequence within the nanopen. And then, of course, you can go to sequence, Sanger sequence, for example, to capture the sequence of that specific antibody that you want to produce then. Once again, fully automated. What you see now here, it's a 20K chip. You can analyze almost 20,000 B cells in a single chip. And once again, fully automated the way that Beacon is able to really select each of these single cells and load them in the nanopen. This is represented here once again. In these different nanopens, you have a single B cell. So what we do then is that we load different types of beads, conjugated beads, to detect the secretion of antibodies. The B cells are here live, they are secreting the antibodies, and so through perfusion, the antibodies will just flow out of the nanopens and go into the corridors. And in these corridors, through diffusion, laminar flow, as I mentioned, you flush in multiple antigen beads, thousands and thousands of antigen beads with streptavidin biotinylated antigen complexes, where basically we can capture the secreted antibody. And then we have thousands of the secondary antibody with flushes to detect that capture. So what you're seeing here in this instant, for example, in this nanopen, you have a B cell that has secreted antigen-specific antibody because it secreted the antibody that did bind to the antigen, and then we see the bloom of fluorescence of the capture of that antibody. Not only that, what you can see as well is that in these other nanopens, you have cells, of course, live B cells as well. Maybe they are secreting antibodies, but they are not actually antigen specific. So you see no fluorescence. More than that, we have years of experience in developing these types of assays. So we have assays that allows you to do cross-reactivity to multiple antigens. So in this case, you flush in anti-IgG beads. And so in this case, we capture the secreted antibody, and it's the antigens that have fluorescence. And so you'll capture, basically, by the bloom of fluorescence, you can see that this specific secreted antibody is specific for this green antigen, but this one is specific for both, and so on. And you can track all of that across time, and Beacon will give you that information. And based on this, you can do other assays. For example, in this case, you have multiple hits. How can you then select the ones that are best? We have multiple functional profiling assays to help you with that. We can rank the hits based on affinity. We can look at functional blocking, where basically in the bottom of the nanopen, we add the target cell to see if not only there's antigen-specific secretion in the corridor, but also if there's blockage of the target cell. Can look at cross-reactivity, internalization that we're about to launch as well. So multiple functional assays. It's not just antibody detection, antibody discovery, bindings, but also antibody profiling and functionally profile those. Then, of course, it's key to get the sequence back. So we can easily retrieve the hits with B cell workflows. Either we can do single cell unloading and getting the B cell, or otherwise we can do on-chip using on-chip cDNA synthesis using our OptoSelect PCR kits, and in this way get the cDNA sequence, amplification, and sequencing. And as I mentioned, we have a team of R&D that has for years now developed on Beacon and developed multiple kits for isolation of from different types of sample sources. Then we have different OptoSelect chips and kits to help you to get the sequence back and as well analyze that data. Plus, what I like to highlight is if the instrument was launched back in 2017, the original Beacon, since then, every single functional assay, every single application, as well on antibody discovery, it's fully compatible with the Beacon. And now that we just launched into 2025 as well, the Beacon Discovery, all of these assays are also compatible with Beacon Discovery, enabling you to do many different types of assays in infectious diseases, autoimmune, and so on. And that's what I'm going to jump now. Some cases just to finish off the presentation. I swear then I will open for Q&A. As I mentioned, we have multiple CROs and pharma using the platform. Some highlights have been the increase in yields when it comes to antibody discovery. For example, from Genovac mentioning an increase by tenfold when it comes to comparing hybridoma to Beacon, especially in hard targets like GPCRs, or the case of Abveris that highlighted really the ultimate flexibility in designing assays, the multiple screenings that you can do. You can just flush in new antigens, screen, flush out, bring new beads, and continue profiling those because once again, the B cells are cultured in the chip and they are alive. And our record is actually culturing B cells for 21 days. So during that time, you can actually do a lot of things. And the same with other immune cell profiling. Of course, it does always depend on the biology of the cell. It's not just in the increase of yields, it's also even in the reduction of costs. Also, s.r.o. in France, in this case, working on antibody discovery as well, they saw a significant reduction of cost when it comes to comparing hybridoma and Beacon because it's almost hands off the Beacon. So while hybridoma took a lot of hours from technicians, on Beacon, it's a lot straightforward, a lot easier to get that data, reducing a lot the cost. And we have as well examples that highlight the power of Beacon on fast screening for antibody discovery. So this case study made Beacon famous actually. It was the work of James Crow at Vanderbilt during COVID-19. So they did have access to one of the first cohorts of patients that did recover, that did have COVID-19 and recovered from COVID-19. So they collected the blood, they isolated the B cells, they screened on Beacon, once again, loading those B cells, then loading the beads with antigens, with spike protein and RBD, and they found antigen-specific antibody secretion and the blocking of the target cell. The full-blown workflow to get those antibodies took them 18 days. What's interesting in this publication is that they did the same workflow at the time together in sequential way, on a multiplex way, at the same time with 10x genomics, and the workflow in 10x took 35 days. Especially when you have to be fast on pandemic preparedness, Beacon really enabled this. And actually, two of these antibodies were licensed by AstraZeneca, and they were part of this cocktail, Evusheld, that was used during this emergency period during COVID-19. And we see other examples, even looking at immunomonitoring of the patient response when it comes to vaccination. In this case, coming from Kate Malasak at Caltech, she wanted to compare two different treatments and survey the antibody responses. And once again, screening multiple antigens. And that's the value of Beacon, is the fact that you can screen multiple antigens and do multiple functional screenings. And she did so to 15 different cross-reactive, and she found antibodies that were cross-reactive to those antigens. But more than that, you can then compare the data because of, once again, linking to the robustness. From first immunization to 4 weeks later, she managed to profile the immune changes based on, once again, these screenings on Beacon, enabling, once again, as well, this capacity to look at even vaccination and immune system response from different patients enabled on Beacon. So since we are close to the hour, last slide, I promise. I wanted to leave you with a message that the platform is quite flexible. I talked mostly about T cells and B cells, but you can load other types of immune cells and look at different sources of functional data, do cell-to-cell interactions, and so on. And of course, do all of this antibody discovery and profiling assays within Beacon that enable to better really find those monoclonal antibodies, especially in autoimmune diseases where you have seen that a very small subset of small subsets of cells that are actually secreting those specific antibodies for those antigens. Beacon has enabled to find those in a very quick manner. And finally, I'd like to thank you so much for your attention. Of course, now I'll open for Q&A. And if you like more information, you can contact me, the Accela team. And also, this is a QR code for our website where we have multiple application notes showcasing some stuff that I just presented today, but also other applications and also testimonials and case studies. And once again, thank you so much. And let's open for discussion. Well, 5 minutes. Thank you so much.
Thank you, Tiago, for the presentation. It was really, really interesting. And I'm
Thank
quite
you, Ricardo.
sure that among the audience there is someone who has some ideas and questions. So before my question, I will give the the
The floor.
space to all the others. Yeah.
Yeah, perfect.
So feel free to unmute yourself as we are not so many, so you can just ask directly, Tiago.
And absolutely, you can unmute, or also if you're shy, maybe you can type it on the chat, or again, you can also contact us
Yeah,
by
I
email.
guess
But
that everyone is
I think they
allowed to
have access
unmute.
to the chat,
Yeah,
right?
yeah, there should be
Okay.
this kind of settings. Okay, so then I will give a couple of minutes to think about, and I will ask you more like practical questions. So about
Yeah.
the workflow and the sample prep. So when you are loading the, I don't know if you were remarking this, maybe I missed that, but when you are loading this chip, you have some, let's say, maximum, I guess, cell concentration, and what's also the timing that it takes to kind of align the single cell using this special system into each of the
Yeah.
channels. So
No,
what's the average
yeah.
time, something like to fill up a cartridge?
That's a very good question, Ricardo, honestly. So in terms of concentration, it really depends on the workflow. On the bottom end, that's something that we have done in the past. You can load really just a couple hundreds to thousands of cells,
Mm-hmm.
because we do know that, especially working on rare samples, it's quite tricky to get samples. So that's a possibility of loading really a low amount of cells into the chip. And just to give you an idea, each of these nanopens has one nanoliter in volume. So the whole chip, if I'm not mistaken, the whole chip has in volume around 125 microliters. Um, so it shows you the precision, the breadth of precision that we have. So we do have standardized protocols in order to load the right concentration depending on the cell type. Especially for B cells, of course, you can load a lot more because you can just load the cells that you want. You can flush away. We can even look at that's very useful, selective sorting in the very beginning. So instead of just blindly loading the live single cells, the ones that are in the corridors, we can load beads with, for example, annexin 5 to look at the viability of cells. So you can do even a pre-sort based on viability, which means that you can load cells, but then you can flush in more cells. Absolutely. You had a second question was, sorry, Ricard, can
About
you?
the timing needed
Oh, the
perhaps
timing.
to load
Yeah. The
fully
timing,
load
it
the
depends on the chip
channels. Yeah.
once again, because of the number of nanopens, but it's around half an hour across the whole chip. Then it really depends on the workflow, the assay. As I mentioned, for example, for cytokine secretion, Beacon will do that overnight, like loading the bits, and so then it's the loading of the immune cells and so on. The time point of the workflow hasn't been a struggle because once again of the ability that we have to culture the cells.
Okay, thanks a lot. And we have a question on the chat box from Tamash.
Yeah.
So he's basically asking if it's possible to additionally add a certain time point in a kinetic way to add a drug in the flowing media and check the effect on the cells.
That's a very good
Yes,
question. Thank
definitely.
you for that question because it also shows you that you're already thinking how to use Beacon. And my response or my reply is yes, absolutely. That's something you can do. I talked very general in terms of what you can do about the assays, but then you can go more complex. And so you have a control over the media. You can also flush in, like, for example, a treatment, and you can look at the response of cells if there is higher activity of the immune cell, for example, against the cancer cell. And yes, the time plot is also changeable. So you can define exactly the time points in which you want really the recording of course, you have the video, continuous video of the bright field of the response of the immune cell, but you can precisely as well look at the brightness points, the fluorescence analysis in a time plot. It can be changed the timeline. So yeah, that's flexible.
Okay, that's great. That definitely a good point.
Yeah,
Thanks, Tamas,
and
for the
good question.
question. And yeah, I don't know if anyone else has any other nice comment for inspiration or question. Seems like no more questions. So we are right on time. Perfect.
Yes.
3 p.m. sharp, and the meeting is recorded anyways. I will just put it together and share it
Yeah.
within a week, within a few days. So for the people who didn't attend or they have some more question, they could have time to review it. And once again, it was really interesting. Thank you, Tiago, for your time and for the presentation, and thanks everybody for the participation.
Yeah. Yeah. Thank you,
Goodbye.
Ricardo, and thank you all so much for attending as well.
Have a nice rest of the day. Bye.
Bye bye.